Okay, so I am picking up our lecture on research methods in biopsychology. We were talking about different ways that lesions can cause damage to the brain. And so we have already talked about tumors and cerebrovascular disorders. So now I'm moving on to concussions. Um, if I go too fast, because I don't have you guys in front of me to cue me into when I'm going too fast, remember that this is a video, so you could just pause it to take notes and then catch up um, with what I'm saying. So the next type of, sorry, I'm on the wrong slide. The next type of lesion we're going to talk about is called a closed head injury. Okay? So this is any brain injury that's due to a blow that doesn't penetrate the skull, right? So you don't crack the bone, nothing goes through the eye socket like we talked about in class earlier. This is when you hit your head hard enough that those meningeal layers can't absorb the shock of your head hitting something, and your brain collides into your skull. Usually this results in what's called a contusion. That's just a fancy word for a bruise on the brain, right? So if your brain smacks into the skull, the part that smacks in is going to bruise. And usually when we see a closed head injury, we see what's called a contrecoup injury, which is basically the idea that the contusion that develops is often on the opposite side of the brain from the side of the head that you hit. So if you can imagine, if you hit something moving forward really fast, so smack your forehead on something, while it's your forehead that hurts on the outside of your skull, your brain smacks into the back of your skull. So it's actually your occipital lobe and your cerebellum where we tend to see the contusion develop. Okay? That's why we get those symptoms that we've talked about of dizziness, kind of like wobbly knees. You see stars. That's because those structures that help with your balance and your posture, as well as vision with the occipital lobe, have been damaged. So this is showing you the CAT scan, which is not something we discussed, but it's taking an x-ray of the brain, basically, of someone with a pretty severe contrecoup concussion. So the front of their brain's towards the top of the slide, the back of the brain's towards the bottom. This was someone driving a car who got T-boned. So they hit their head really hard on the left side on their window, but you can see that it was the right side of their brain that smashed into the skull. And what you see there, what the arrows are pointing to, is an inflammatory response. When that contusion develops, the glial cells help trigger this immune response to bring in cerebrospinal fluid to pool around that damaged tissue to just kind of protect it and make it a little bit less likely to hit the skull again while it's healing. So in the short term, this can be a good thing. But in the long term, we need that fluid to eventually drain away because, of course, it's putting pressure on the brain. Right? So when we say somebody has water on the brain, or that there is swelling on the brain, that's what I'm trying to say, this is what's happening. I don't know if you guys remember back when Hillary Clinton was Secretary of State, she fell and had to have surgery to drain swelling off of her brain because she hit her head pretty hard. And so this is what was happening to her. She had this inflammatory response that needed to be drained out because it was too much, creating too much pressure on her brain. Of course, the most common type of closed-head injury is a concussion. I'm sure if we were in class and I asked people to raise their hand, we would see that several people in class have had concussions. We classify a closed head injury as a concussion when there's some kind of disturbance in consciousness. Now, that doesn't mean that you have to lose consciousness. You don't have to black out for it to be a concussion. anything that changes your current perception of your experience is changing your conscious experience. So for some people, they might report that they feel like time slowed down for a while. Other people might just report having difficulty remembering what happened for a period after their head injury. So anything that kind of alters your consciousness, alters the way you're thinking or processing information, can be classified as a concussion. And as you know, we are learning that concussions are much more serious than we used to think. Scientists didn't used to think that concussions were a very big deal because if we do an MRI right after a concussion, we won't really see any dramatic brain damage on those kinds of scans. But that's because when you have a concussion, it basically, when your brain smacks into your skull and causes these shifts in consciousness, those shifts are a result of new synapses that have grown breaking off. Right? So there might be some kind of new information you've learned, or maybe even representing like a mood state or a feeling that you've been having. And the synapses that represent that information get damaged. And we can't see synapses in the brain imaging techniques that we have right now. But now we're learning people who have multiple concussions or even one severe concussion can eventually result in the development of dementia. That basically so many synapses get broken in this process that there's confusion and memory loss, much like you see in Alzheimer's disease. Colloquially, this is referred to as punch-drunk syndrome. That comes from the study of this phenomenon in boxers, where people who box take a lot of blows to the head. They usually experience multiple concussions throughout their careers, and typically die earlier than the average life expectancy, and die with full-blown dementia. If you guys have ever heard Mike Tyson talk in recent years, you know exactly what this looks like. All right. So concussions are another way where we can get lesions on the brain. The lesions are just so tiny that you can't image them. We can also get infections in our brain. So this is when some kind of microorganism gets into the brain space. Often this causes encephalitis, which is again inflammation that is an immune system response to this microorganism being in the brain space. So again, you can get swelling on the brain in response to this. So brain infections can come from bacteria. So this, just like if you get a bacterial infection anywhere else in your body, this is going to lead to abscesses, which is pussy pockets, which of course are going to damage neural tissue. Again, you can get inflammation in that meninges layers, creating meningitis. And just like with anything else bacterial, the good news is this, that we have antibiotics and penicillin and things that kill it pretty effectively. So we've knocked a lot of the bacterial infections out. So for example, syphilis, a sexually transmitted disease, is a bacteria that can attack neural tissue. But we've treated it pretty much to the point where it's no longer present in our population. But bacterial meningitis, I think I mentioned earlier this semester in class, is an infection that does still exist, and actually one of the most common places we see it is college campuses. Because when this bacteria gets into dorm spaces, it spreads really quickly, and it's really unpleasant, but treatable, so not as awful. Viral infections, on the other hand, are not as great. So there's a lot of viruses that preferentially attack neural tissues. For example, rabies. If you contract rabies from an animal bite, there's really no treatment for that, right? That's why we do vaccinations against the chance of having rabies. So you can prevent it from developing, but if it develops, there's not really a way to stop it. And there's strands of the herpes virus that also can selectively attack brain tissue. All right, so those are ways that brain infections can happen. Um, we also have neurotoxins. So these are things that enter our circulation through our gastrointestinal tract. So we eat them, drink them, breathe them in through our lungs, or just have contact through the skin. So anything that basically can get absorbed into our circulation and the result of that is damage to neural tissue is considered a neurotoxin. So often neurotoxins cause what we call toxic psychosis, which is a chronic and untreatable insanity produced by the neurotoxin. So we have some kind of phrases in our society that refer to some of these. So the Mad Hatter from Alice in Wonderland, that phrase, somebody says mad as a hatter, comes from when hatters used to felt wool to make things like fedoras and those kind of hats. And they discovered that if they used liquid mercury to rub the wool, that it felted really fast, so that means it just got nice and soft really fast. And that was before they knew mercury was a neurotoxin. So mercury you can absorb through your skin, or as it evaporates and you can inhale its fumes, it destroys brain tissue. Right, so that mad as a hatter comes from people going crazy from their brain tissue being destroyed from exposure to mercury, specifically people who made hats. The phrase crackpot also comes from the same time period where they made teapots out of lead and they would dip them in porcelain. So you had porcelain-coated lead as your teapot. And if you were wealthy and your teapot cracked, you would just throw it out and buy a new one. But if you were poor and your teapot cracked, you just kept making tea in that pot. And so the lead that was inside the porcelain coating would start leaching into your tea, and we now know lead is a neurotoxin. And so poor people were going insane from exposure to lead that they were drinking in their tea. Right? So this is why people freak out these days when we discover that some child's toy from China or somewhere has been painted with lead paint, because as kids tend to put everything in their mouth, they're exposing themselves to lead, which can cause brain damage. All right, so just a couple examples of environmental toxins that can destroy brain tissue. All right, and then just have a couple more examples of different types of lesions. So this is a MRI image from a patient with multiple sclerosis, right, or MS. So what you can see is that this really bright white region just above their ventricles is where there should be healthy tissue for the corpus callosum. But instead what is happening is that the myelin on the axons for this area of the brain are calcifying and becoming scar tissue. Right, so that's not healthy functioning brain tissue, that's now calcified scar tissue. So neurons can't communicate using these axons anymore. Right, so multiple sclerosis, we don't have a definitive cause for. It's been linked to things like your vitamin D levels. There's also seems to be a genetic component. But it basically triggers this malfunction of oligodendrocytes where they're no longer maintaining the health of myelin, and that results in the lack of communication among these damaged neurons. Right? So that's where you get the motor movements, the motor symptoms that are common for multiple sclerosis, but you also start to get cognitive symptoms as well. Right? There's often memory difficulties, confusion, and similar symptoms. As well as like the fatigue, the muscle fatigue, losing coordination with movement, and that sort of thing. All right, and then these two slides are from the neural tissue of a patient who died and was definitively diagnosed with Alzheimer's disease. So until recently, you couldn't 100% diagnose someone with Alzheimer's disease until they died, because the thing that makes Alzheimer's unique is microscopic. So again, you have to be able to put their brain tissue under a slide, under a microscope on a slide, in order to diagnose the disease. Now we're starting to learn that you can detect some of the proteins associated with these things in their saliva and in their bloodstream. So we're getting better at definitively diagnosing Alzheimer's disease while people are still alive. So what we see here on the top left of your screen, that is called a neurofibrillary tangle. So it's basically a tangled mess of fibers that starts appearing in the extracellular space around neurons. It basically just gums up the ability of neurons to communicate with each other. And then on your bottom right, we have what's called an amyloid plaque. So just like the plaque on your teeth, it's a plaque that also develops in the extracellular space. Again, interfering with the ability of neurons to communicate with each other. Right? So when neurons that used to be connected and be able to communicate with each other can't do that anymore, that's where you get confusion, memory loss, because the information they used to process and represent isn't being represented correctly anymore, right? There's going to be mistakes made because neurons aren't firing and communicating in the same way that they used to be able to. All right, so that's a whole bunch of different ways that we can look at lesions in the brain. But some important things to know whenever you're looking at lesion research is that the data is going to be really messy, right? You can't control who your participants are when you're doing lesion research, right? You're not going to decide who gets Alzheimer's disease. You just have to take whoever has that diagnosis, and so there might be a lot of third variables that are influencing your data. But also, even if you have a group of patients all with a very similar lesion, the likelihood that there is 100% overlap of their lesions is pretty much next to nothing. All right, so what this slide is showing you is 4 patients who had similar strokes. And so each patient has their MRI slides, and the red shows you the extent of their strokes. So on patients 1 and 2, the stroke was on the right side, and on patients 3 and 4, the stroke was on the left side, but on an analogous structure, right? So the same part of the left side as was damaged in the right side in the other 2 patients. So in this study, what they did is they mirrored the damage of patients 1 and 2 so that all the damage was on the left side. And then they made this composite of the damage shared by these patients. So in this composite, the color red means all 4 patients had damage to this area. The color green means 2 of them did. Green means 3 of them did. Blue means 2 of them did. And purple means only 1 person had damage to that area. So you can see if you're trying to infer across these patients that damage to this part of the brain results in a specific behavior or side effect or symptom, it's going to be difficult to say that that one spot that they all had in common is the only thing contributing to their symptomology, when there's a whole bunch of other parts that got damaged that differ between all of your patients. Right, so basically you just get really messy data when you're doing lesion studies, so you have to be really careful again with how you apply those findings, right? Your external validity is not very large when you're doing this lesion research. Okay, so the pros and cons of lesion research. The pros is that it's the ultimate test of neural function, right? If you really want to know if a part of the brain is necessary or not for some kind of function like speaking or learning, and you see someone get a lesion to that part of the brain and that function goes away, you know, okay, that part of the brain is critical for that function. The cons are that, again, the damage is often widespread, so it's hard to have really clean, neat data when you're looking across patients. You can only study what you get, so again, you can't randomly assign your participants. You can only study the people who already have that damage. And then there's two kind of logic errors that people tend to make when interpreting lesion data. So the first is making a disconnection error. So that's when if you have areas A, B, and C, and all are involved in some kind of process, let's say talking for this example. And someone gets damage to area B and no longer is able to talk. And so you might infer that, oh, area B was where talking was created in the brain. But it could be that area B was just a relay station, just a part of the brain that passed information from A to C, and C is actually the part of the brain that creates the behavior of being able to talk. Right? So a disconnection error is assuming that the part of the brain that was damaged did that function specifically, when it could have just been a relay station where that information had to pass through in order to get processed. All right, a compensation error is an error you make by not taking into account the brain's ability to compensate for damage. So in this case, if you have areas A, B, and C, and area A gets damaged, again, let's use talking as an example, and the person is still able to talk, you might infer that A was not necessary for the ability to create language. But you have to take into account that the brain compensates very quickly, that a secondary area can take over the job of an area that's damaged, and it can start doing that within hours of the damage occurring. Right? So usually in a research setting, if somebody has a stroke and goes to the hospital and is treated, you don't get to go in and research them within hours of that stroke happening. Right? The primary objective is to make sure they're stable and healthy and recovering. So maybe several days, more likely several weeks after their stroke, you might get in there to be able to study them. And that is a lot of time for other neural regions to start compensating for the function of the region that got damaged. So just because they're still able to do something doesn't mean that the brain damage they had wasn't important for that function. It just means that compensation has already occurred by the time you've studied them. Okay, our last methodology is transcranial magnetic stimulation, or TMS. There's two YouTube videos that I would have shown in class for this, so I'd like you to check them out on your own. So how this works is it's again based on an electromagnet. So these two circles are strong electromagnets. So this is able to project a very strong magnetic field into the brain. And unlike an MRI where it was like a huge magnetic field across the whole head, this is really targeted. So we can target areas of about a centimeter with this machine and only influence those neurons. So you can basically manipulate the magnetic field produced by a TMS to either stimulate a part of the brain, right? So you can turn a part of the brain on and see if that changes a person's behavior, or you can manipulate it to inhibit a part of the brain and basically stop a part of the brain from working temporarily. So you can create these artificial lesions that go away as soon as you turn the machine off, and you can test to see what happens if people don't have a part of their brain working. Okay, so the video will show you some journalists who are manipulating Broca's area, which is the part of the brain I'm using right now. It's the part of the brain that helps you produce speech, right? Allows you to talk. And you can see what happens when they are doing that. So with TMS, it's relatively inexpensive, so kind of like EEG, it's accessible to a lot of people. It lets you do artificial lesions on humans, which is the only way we have for doing that on humans. The cons is that it's only really valid for cortex. So you can imagine if you're using that technique and you want to like turn off the amygdala, the only way to do that is to also turn off the cortical tissue in between the skull and the amygdala, right? Because you have to put the magnetic field in from the skull. Right, so if you want to look at subcortical functioning, it's hard to isolate the effect that you're having. It's also a pretty new technique that's being used in research. It started becoming popular in the early 2000s. So as far as we know, there's no side effects to having TMS procedures done on you. But it could be that in 50 years we find out that people who were TMSing regularly might have some kind of long-term effect. Again, there's nothing to really suggest that that might happen right now, but it hasn't been around long enough that we could say it definitively. You can, at this point, get on Amazon and buy your own home neurostim kit that basically is using much, much, much weaker magnetic fields to mildly stimulate parts of the brain. So you can check that out. I have no idea how valid they are, to be honest. There hasn't been a lot of great research that have been done on them. Alright, so to wrap up this chapter, there are many methods for studying brain function, and each has a different contribution to make. So understanding the brain will require putting all of these different puzzle pieces together. Right, so remember, for each methodology, I highlighted pros and cons of each. So each are really appropriate for some types of research questions, but aren't as great for other types of research questions. So kind of understanding the applications and which methodologies are better for which kind of questions is an important take-home here. And also just understanding what kind of signals they measure. Alright, so that wraps up this chapter, guys. And I will hopefully get the next one up for you guys soon. Bye.